We would like to thank the editors of Endoscopy for the opportunity to discuss our new paper entitled Microbiology of Bile Aspirates, obtained at ERCP in patients with suspected acute cholangitis. My name is Mark Gromski, and I'm an advanced endoscopist at Indiana University School of Medicine. It's my pleasure to be here with my colleague and my co-first author of this paper, Dr. Aditya Gutta. Aditya, can you please let the listeners know what was the impetus for this study?
Thanks, Mark. Over time, in the patients we've been taking care of, we anecdotally appreciate a change in the microbial pattern and specifically an increase in the antibiotic resistance of organisms causing ascending cholangitis. Over 20 years ago, we published a study that described the microbial composition of biliary aspirates for patients with suspected cholangitis. Since that time, we found it to be very useful to obtain bile cultures to help us guide our treatment of these patients, and thus we routinely aspirate bile in patients undergoing ERCP for suspected acute cholangitis. We sought to update our knowledge and data in this area by determining the current microbial pattern and drug resistance for patients undergoing ERCP for this indication.
Thanks, Aditya. In this study, we analyzed biliary aspirates from 2010 to 2016 that were obtained at the time of ERCP for suspected cholangitis. 721 ERCP procedures were performed during that period that met these inclusion criteria. Of these, 92% had positive bile cultures, and of the 437 patients with concurrent blood cultures, approximately 40% of those were positive. Aditya, can you go into some further detail of which organisms we found?
Of course, Mark. Of the 662 cultures that had microbial growth, 81.6% were polymicrobial. The most common pathogens were Enterococcus, Klebsiella, and E. coli, which grew in 68%, 45%, and 41% respectively of positive cultures. But many different bacteria grew in smaller proportions of the positive cultures.
Thank you. Now, in our institution's prior study, we showed that there's a difference in the microbial profile for patients who had an indwelling biliary stent, with higher incidence of polymicrobial culture and more prevalence of Enterococcus. Although similar and likely related in mechanism, there's less known about the microbial pattern of patients with cholangitis who have had a prior biliary sphincterotomy. Can you share what our study showed in those regards?
Yes. So our current study confirms the effects of a prior biliary sphincterotomy was similar to the microbial growth of patients with a prior indwelling biliary stent. A prior biliary sphincterotomy was associated with an increased risk of Enterococcus, Enterobacter, Pseudomonas, and Klebsiella species in this study compared to patients with no prior biliary sphincterotomy. And these findings were similar to the cultures from patients with indwelling biliary stents.
Another one of our objectives of this study was also to determine the antibiotic resistance patterns of the bacteria that we found. We found that the Klebsiella bacteria were susceptible to ciprofloxacin in only about 88% of samples, and the E. coli was only susceptible to ciprofloxacin in 64% of samples. These are higher resistant rates than our prior study that was performed over 20 years ago in a similar cohort of patients, and thus likely represents increasing community resistance to this commonly used antibiotic in our area. Aditya, we've heard a lot in recent years of multi-drug resistant bugs such as ESBL, MRSA, and CRE. Can you share how many of those organisms we found in our cultures for patients with cholangitis?
Yes, we'll be glad to. We did find some of these multi-drug resistant organisms that some term as superbugs. Among the Enterobacteriaceae group of organisms, we found that 8% were ESBL and 4% were CRE. Among the Enterococcus isolates, 15% were VRE. We were somewhat surprised with the high prevalence of these organisms. When applying the subgroup analyses, we found a prior biliary sphincterotomy significantly increased the risk of VRE.
It's very interesting. This is a compelling reason for why endoscopists should consider aspirating bile for patients with ascending cholangitis. In these cases of resistant organisms, it helps to direct treatment promptly with the most appropriate antibiotics. Over time, it also provides an antibiogram to the institution to help decide the optimal empiric antibiotic regimen to treat patients with while they're awaiting biliary decompression. There has also been quite a bit of debate and discussion recently of the potential for transmission of resistant organisms through our traditional reusable duodenoscopes. At our center, in patients who require subsequent ERCPs, such as patients with primary sclerosing cholangitis or a hilar stricture, We also utilize historic bile culture results to help determine and decide whether we should use a single-use duodenoscope. In patients with a multi-drug resistant organism on prior bile culture, they fit into our top-tier category to use a single-use duodenoscope. Dr. Gutta, I'd like to thank you for joining me in this discussion, and again, thanks to the listeners and to the editors of Endoscopy for the invitation.
Thank you, Dr. Gromski, and thank you, editors of Endoscopy, for providing us a platform to amplify our research.